Thank you, Dr. Roll, for that great talk. I'd like to invite Dr. Gremez from Sandoz to kick off the next session. Thank you.
Hello, good morning. Good morning to everyone. So it's both a pleasure and honor to be here, to have opportunity to present one, let's say, real case study from the industry. So yesterday and also today, we heard a lot about the expectations from the particle sizing methods, about the accuracy, about the precision, other expectations. But in the end, I think we all agree, first thing that we need to understand is a purpose. And I hope that what we discussed yesterday and today, let's have this in mind while we go through the presentation. Thank you. Okay, so the plan for today. First, I will briefly frame the need, the purpose, which turned into a challenge. Of course, with the challenge, we need to find solutions. There are probably many solutions, but I will present two of them. And in the end, I will conclude with the unmet needs and hopefully also provide some cues for the panel discussion or maybe some offline discussion during the break. Okay, let's first talk a little bit about the purpose, what we are doing. Okay, we have a goal to develop particle size distribution method for in vitro bioequivalence purposes. What is our product? We have a thixotropic gel suspension with the API in the submicron range. We can all agree that this is a complex formulation and goes well in the scope of the forum where we are. The requirement, the method needs to be robust. We need to develop for the in vitro bioequivalence. So for those who are not familiar, this is a formal comparison, which is usually done by comparing at least 3 batches of test and 3 batches of reference product. For each batch, not less than 10 real replicates are done. The requirements and some other guidances are usually written in the product-specific guidance. And in the end, this results in, let's say, at least 60 measurements. Out of these 60 measurements, we compare defined metrics, in this case, D50 and SPAN, by using population bioequivalence statistics. What's important for the formulation? It's a complex formulation. We don't want to alter the formulation, since this can also temper what we are measuring. So the API, meaning that a lot of sample preparation steps, we need to keep any manipulation at the lowest possible level. Also, what's important, the method must reliably measure particles below 1 micron. We've heard yesterday and today the DLS method, preferably below 100 nanometers. LD method above 1 micron, meaning that with this product we are hitting exactly the gray zone where neither of the methods that are available are perfect. Still, let's say out of the available methods, what were considered, electronic microscope, out of question, because we would really have to manipulate the sample, and also in terms of statistical power, it's hard to collect enough data. Optical microscopy, we are limited by spatial resolution, meaning that also with 0.5 microns, considering that this is probably the median of our range, it's not good enough. Raman microscopy, still limitation of optical microscopy, but it would come in hand, for example, if we would have several different components, so there is no major advantage here. While laser diffraction, even though it's not material specific, it's still the golden standard in the industry, meaning that this was also our decision. Not listed here, but we also discuss DLS. So due to the intrinsic nature of the DLS method, Brownian motion, probably we would have to manipulate sample to the very severe extent, meaning that this would also probably impact the results we would read out. So our choice was laser diffraction, and we also choose optical microscopy as supportive method. Why? We will see on the next slide. So I think so far, this is the decision process. Let's start with the measurement. And when we did this, this is what we got. So we got unexpected bimodal particle size distribution. If you refer to the figure 1, you can see that there are two peaks. The major, the big one, and there is a smaller one. Based from the understanding from the product, we did not expect this. So what are the possible root causes for this? Maybe some agglomeration happened, something else, but this is something that we are not that it's not possible to see with the laser diffraction. This is where we can get the results from optical microscopy, which indeed confirmed that this large peak, so at lower, smaller PSD values, really belongs to the API. We did not observe any agglomerates, and the larger PSD peak belonged to the excipient, meaning to the placebo. The good thing is that even if not visible here, the peaks were very well separated at the baseline of the histogram. So one tempting solution would be, let's simply cut it off. But what's important, we needed to understand more how the method behaves. So we went into the analytical design of experiment. Many different things were tested here in the figure 2. Some of the experiments are presented, and we were able to, let's say, modify the signal of the placebo, while we found also conditions where these changes did not have any impact on the API peak. So this was good. We were able really to suppress the placebo signal to the highest possible extent, but still this was not sufficient to completely remove it out of the picture. The placebo peak was still there, meaning that it impacts directly your measured results. So the challenge was, how can we isolate the API signal? Two approaches. First, I would just like to explain the right part of the slide, because it will be the same for the next slide. So on the top, what you see is overlay of one typical sample using by approach 1 here and approach 2 on the second slide. So this is what we would typically see when the production process is giving us robust product. On the image below, you can see where the production process did not go well, meaning that the agglomerates appear. And the purpose of this is also to demonstrate the worst case scenario that could happen. You can see the overlay, so meaning that the both approaches showed the similar story. Now, let me go back to the left side. So, with the first approach, usually when you measure with the laser diffraction, as part of the procedure, according to the manual USP, you also need to measure background as a blank solvent-free medium. However, the group from An Vo and Tol really developed an ingenious and novel approach where they used placebo instead of the blank solvent. And this was then subtracted by the measured sample. What's important with this approach? So it's in a way, it's a little bit of a black box. So you are removing something directly from the raw data. So you get out the subtracted histogram, meaning that something is subtracted, placebo, considering that placebo particles are also in the formulation, meaning that this is the results that we get. Also, what's important due to nature of the method, since we are directly adding this product for the measurement into the wet dispersion cell, is also what we are adding it and to what amount we are adding it, meaning that also the composition, concentration, and the amount of placebo needs to be comparable to what we put into the for the sample measurement, meaning that the results are also highly dependent on the how well you control the experiment. If you don't control it well, then you will introduce additional variability coming from the method. Nevertheless, the result compared to the approach 2 seem to give similar outcome. For the approach 2, as I already said in the beginning, it's pretty simple. What we did, we defined the lowest area between both peaks, and we simply did the cutoff of the higher peak. So what is the advantage of this approach? We still measure the raw data, so original data contain the placebo peak. Only after original data is obtained, we remove the placebo peak, so we can always go back and see what the original data looks like. And what is the major disadvantage? Of course, it's a non-specific cut. What we do is simply we have a look at the lowest point, and even though it is on the baseline, what if there are some particles in the area above that, so hiding under the peak? This is probably the common issue for both of the methods. And of course, this method works only if we really can confirm that these particle populations are clearly separated. And now the big question, which approach out of these two is more scientifically sounded? Well, that really depends on the case. So I presented one specific thixotropic suspension. There are many other cases where peaks are less separated, more separated, some other factors from formulation which can impact your decision. Nevertheless, we don't have clear answer, but the good thing is that using either of these approaches gave the same result. Okay, I'm coming to an end. So basically, as I already explained, each of the approach has some assumptions. For the placebo subtraction, you need to know the placebo composition. As generic, we know it for our product, we assume it for the reference product, but let's say we can safely assume it. Still, there is a subtraction algorithm which needs to be considered. For the second approach, of course, you have to know that both populations are clearly separated. And these assumptions, in this case or in general, they cannot always be validated. But what's important, whoever develops the method needs to provide this scientific justification on why this was selected. In this case, it's not only about the method development, but it's also about the understanding of the product, of using orthogonal methods, really to show, for example, are the particles above 3 microns, where we have the cutoff value, really present in the formulation? Can we expect them? How can we detect them? So this is all the work that needs to be included in the method development. To finish, for the panel discussion, what I would like to maybe set some ground is for the instrument manufacturer. I already asked yesterday what are the advances in terms of new techniques. How can we do this? How can we get better, more reliable measurements? For the colleagues from the industry, this is the forum where we can share these practices, how we do things, what are the challenges. And of course, for the regulators, we also heard yesterday the need to maybe provide maybe some more specific instructions in terms of, let's say, direct guidances or references or something else. So with this being said, as I said, it's not only expert for the method. There is a whole team behind, from pre-formulators to formulators to analytical in vitro B experts. Each provides necessary input to define the purpose, and from this purpose, we are then able to build this story. So my thanks go to them. They're my motivation to come to work, and honored also to work with them. With this, I conclude. Thank you for your time. This is it.
Thank you, Dr. Gremes, for that wonderful talk and sharing all those core challenges and the needs of the hour. Certainly insightful for CRCG. With that, I'm going to introduce Dr. Rama Subba Reddy from Dr. Reddy's to give the next presentation.
So good morning, everyone. So my name is Rama. So I'm on behalf of Dr. Reddy's Laboratories. So first of all, thank you very much for conducting this wonderful workshop. So where I think all the industry experts came together to discuss about the challenges and advances in the particle size analysis. So it would be very interesting to see that follow-up workshop on the what are the challenges we discussed, and so how like useful for the industry and academics. So with this, let me start my talk. So I'll be presenting two case studies. Mostly first study will be like in line with the what just now we saw from the Sandoz. So yeah, case study one will be about the challenges in developing a DLS method for the cream formulation. Second one will be the self-association impact of the protein peptides. So what is the heterogeneity in DLS? Let us define that. So as we, I think, yesterday discussed already about this. So fundamental assumption of the DLS is the particles will be in a spherical shape, and most of the times it will be like a same size, same shape. So with a narrow, like a polydisperse index, we call them as a monodisperse. But however, in real case scenario, no formulation will be that simple. So you'll always be having a broad distribution, different shapes. So this is called as a polydisperse. But so one step ahead, we will be having a heterogeneity issues, something like this. So where I think your formulation is not a monocomponent, like a multi-components will be there. Along with that, you will be having a lot of oil globules, micelles, all those things when it comes to the cream formulation especially. So I'm going to talk about this heterogeneity, how it is impacts the method development, what are the key challenges we are facing. So let me go through the case study one. So the data which is coming from our lab, so which is all like mostly what we generated. So before going to the globule size distribution, let me like what a lot of the people discussed about the orthogonal techniques. I'll present what I understand this product from all the orthogonal techniques. Yes, this is a cream formulation. In light microscopy, I'm not seeing any visible particles there. So it's mostly in a nano. Then, so when I turn this into the polarized microscopy, I am seeing a clear anisotropic domains. That means some clear structure is exist in that cream formulation. So let me further probe the structure of this cream formulation. We have implemented like a SAX and wide angle X-ray diffraction studies. So from the SAX data, I think you all be most of the people understanding that from the periodicity, we can clearly see that there is a clear structure exhibit. So it could be a lamella. It could be a clear lamella. So then the question is like a cream formulation, which is having a lamella, whether do we really need to develop a DLS method or a GSD globule size method? So let me explain here. This is all about the innovator data I'm presenting, what we analyzed in the lab. So innovator clearly having a patent on the globule size distribution. So it's like IP perspective also, I need to like define what is my filing, like it's a para 1, para 4, something like that. So I have to develop a method for the cream formulation. I need to show that I am not falling within the, or within that patent. So let me go through the GSD data. So this is a sampling, how we have done. So we have taken a 10x diluted sample in the water and then sonicated for 5 minutes, and then DLS was analyzed. So from the intensity, I am not seeing that much impact, but however, in volume, clearly I can see that multiple populations exist, and these are like a real, when we repeated for multiple times also, we are able to reproduce this same data. So we have gone back to understand what are these 3 different peaks. So we have conducted a cryo-TEM analysis. From that, we clearly see that I think the dark spots which you see is the one from the phospholipids, and the transparent globules, which is from the oil droplets. So there are multiple populations clearly exist from, this is holistic understanding on this project. Then we need to develop a GSD. This is really interesting. So then we understand that the lamella which we are talking about is not that much simple. It's not just a monolayer. It could be something like this. So it's a lamella along with the micelles, bicelles. It could be in between the lamella or outside of the lamella. Or maybe I'm creating during the sonication of my sample. We don't know. So this is very, it turned out to be very interesting. Then we have gone ahead to understand what is the impacts, all we discussed about the dilution impact, sonication impact, sample concentration impact, and the stability of the solution. So everything we have done everywhere, I think we could not able to understand or to develop a robust method. It's always coming like a multiple peaks, and how do we set up a specification, all those things becoming a very challenging. So these are some of the questions which I framed for this product. So how to finalize the sample preparation for this such kind of a cream formulation? So as I said, it's really required because patent is there. And then further, if I match only my SAX periodicity from test product to RLD, is it okay in the aspect of the agency? If we want to prove that bioequivalency or POB, so how do we go ahead, like earth mover distance, or is it okay to do with any statistical approaches, INNOVA or PCA? So then the last question is like a setup specification, 3-tier spec, usually the agency expectation. So how do we set up this? So this is the case one, and then let me go through the second case, where I think this is, most of the people's favorite, carrier and plasma protein self-association. So to explain this, I'm taking an example of the phenomena called protein corona formation. So what is a protein corona formation? When you have a nanoparticle which is subjected for the systemic circulation, so what are the, in blood, you will be having a HSA, you will be having a BSA, you will be having a PF4 platelets. These all interact with your nanoparticle. It will try to create some kind of a complex, or maybe your nanoparticle will get dissolved. This is some kind of a complex phenomena will happen. So this, we will do it for most of the nanoparticle to understand in vivo performance. In OSD molecules, you can see that people will run their resolution in the passive, biological relevant media. So similar way for nanoparticles, we try to understand, so what is the impact of these nanoparticles when we really give it to the patient or going before for the bio studies. So this is called a corona formation. So in this, what will happen most of the time, so these protein or carrier proteins, so usually have a tendency to form a self-aggregate. So that will really screw the data, and it will really have a hard time to interpret the data, setting up the understanding whether it's corona formed or not formed, all these things. So this is the holistic understanding on the phenomena, and I'll present a case with the HSA, especially HSA, so how it will impact. So HSA in general will come with a monomer, and aggregate percentage will always be there. So this is the one of the lot of the HSA, human serum albumin. So you can see that 66% of the monomer or dimeres exist, and then there is a 34% of the some kind of aggregate is existing. So when we take, so you can see that we have, I think I highlighted the count rate there. So since it is a bimodal distribution, it is always better to go ahead with the count rate instead of the G average or distribution analysis. So we have like plotted the like count rate against to the different concentration of the product. So what we have done, so we have taken the blank, blank HSA, which is at a blue color, followed by we have done a spiked the drug product into the HSA with some low concentration. We can see that dissociation or dissolution happened immediately, which is matching with the blank solution. However, when we increase the little bit more concentration, we can see some kind of a kinetics happening. It is taking some time and then getting dissolved. And then with a high concentration, which is a C3, which is not at all dissociation, which is, I think, already reached the saturation. So this is the like a textbook kind of a picture where people will generally try to get similar kind of a result. But what happens when you change the lot? So there, I think I'm having a more aggregate, the data getting all around like screwed. So my baseline become a very high. Still, in C2, like a medium concentration, I'm seeing a dissociation, but the kind of response I got for the lot 1 and the lot 2 is like varying differently. So now, so with the same product, I have done with the 6 preparations for the C2, and you can see the clearly the how this slope and dissociation is like changed. So above one is with the first lot of the HSA, where I can clearly see the dissociation and all six triplicates are matching with each other. And then we have gone ahead with the second lot, and we understand that the dissociation slope, everything got changed. Now, when we apply statistics to this, it's not at all meeting the criteria of the agency. So this is one of the very hard time. And then similar case with the PF4. PF4 usually, I think, used for this agency requirement is from the, when you worked on heparin-based molecules, you need to show that I don't form any like extra complex, which is called heparin-based, heparin-induced thrombocytopenia. So HIT, when you interact PF4 with the drug product, there is some concentration, very high large molecule will form. Like, so it is called as a HIT. So from the agency expectation, drug product as well as the RLD should form the similar way, similar size of the ultramolecular, ultra large complex. But PF4 itself is coming with a lot of challenges. So as I clearly mentioned there, single tetramer is the one which is responsible to form an ultra large complex. Aggregate will not be interact with the drug product. So here interesting thing is what lot of commercialized PF4s are comes with a lyophilized powder. So when you try to disperse them with water, it's have a auto aggregation tendency within the water. So you disperse with water and keep it there for some time. When you come back and analyze, it will automatically single tetramer turns into the aggregate. So this negative experimentation, you may feel like, yes, some complex is forming, but blank itself is converting into the aggregate. It's not actually any interaction is happening there. So you need to screen as dispersant where your PF4 is very stable, and followed by you need to conduct the experimentation. Unfortunately, the literatures available over the internet, most of them, the PF4 is dispersed in the water. So no one talked about the stability of the dispersant. So I have 2 lots of the PF4. When we analyze that, I can clearly see that there is a difference in the single tetramer ratio. So this is impacting a lot on my size, which is coming out of my ultra large complex. So this is when we submit the data or when we submitting, giving for the review to the regulators, everyone question is like how we can justify this. So these are some questions which I framed, like how to overcome the lot-to-lot variations, selection of the dispersion for the stability, instrument impact, especially the attenuator. So most of the times we'll try to keep the attenuator as auto, but when we run the instrument, different concentration, you cannot like fix that attenuator. You need to keep them as find out the better response for the sample. So these are the things like maybe I'm looking forward to discuss at the panel discussion with all the industry experts or agency, how we can overcome this kind of a thing. Thank you.
Thank you, Dr. Reddy, for sharing those case examples with us. I'd like to invite Dr. Mr. Dominic to, Dr. Ayub, yeah, to provide the next presentation. I apologize.
Good morning, everybody, and good afternoon and good evening for those who are joining us online. I would like to start by saying a big thank you for all the people who worked to make this workshop reality. I know that took more than 2 years, and already yesterday, after I think the first or 2 sessions, it is clear that the 10,000 kilometers that I passed to come here was really worth it, so thank you so much. I will speak about disclaimer. I will go a little bit fast because I know that we are behind schedule. So we all know that PSD is a critical quality attribute. We all know that laser diffraction is one of the most used methods. So I will go fast to the main 3 topics I would like to discuss today, which is basically gaps that we have found in when we go through and analyze the big number of questions that we have with the authority. Teva is around in generic industries around for more than 40 years. So we have more than something like 35,000 questions and answers with the authorities. And we usually do analysis. We used to do that manually. Now we do it with the assist of AI. And we located few topics that is worth stopping next and dealing with. One is the validation gaps. And the validation gaps is basically 2 topics, which is the accuracy. There are different approaches to perform accuracy for laser diffraction method. And we surprisingly find that different sites within Teva is taking different approaches. Some of them are already doing this for years, but we did not know that each site is doing different accuracy. I will speak about this in a minute. And the main problem was that several authorities came back and asking accuracy using microscopy. And I think it was raised today that microscopy is a 2D and we are working with 3D, and the challenges were notable in this part. The second thing I want to speak about is setting specification. I think we have raised this yesterday. Setting specification is one of the main challenges, especially in the generic industry, because we start the development in many cases when the drug substance is almost in the final stages of their development, and then we only have 3 batches to go forward with. The 3 batches will be part of the clinica. The 3 batches will be part of the validation of the method, and these will be also the submission 3 batches. So we have 3 batches to set specification. And I think Alan said that very nicely. When you have 3 samples, three samples is not good enough for statistical setting specification. But this is what we have in hand, and this is what we need to go forward with. And in generic industry, time is the most important thing. You cannot wait for the 4th or the 5th batch. And the last thing it was really discussed is the technology transfer. There are sites who have decided not to participate in technology transfer. They say, just send us the method. We'll take the method. We'll develop our method. We'll validate the method from scratch, which is easier for them instead of performing technology transfer. Not because the method is not robust or something. That's because different technology, different people. We have even found cases where different humidity in the different places will affect your result. So what we did in the last few years within Teva, we have developed an AI based in natural language processing, which is an AI that can understand human language. And we went through all these more than 35,000 questions, and each one of these questions has tens of pieces of information. So in general, it's more than 300,000 pieces of information that we search within. And using this, we have located the main painful point. And for the first cycle that we did this analysis, it was accuracy. For 3 years, 9 out of 9 questions related to particle size laser diffraction, the word accuracy came in. And we understand that authorities are also not sure how they need us to do accuracy, but they are asking us in different ways. So yeah, the FDA is basically doesn't have a specific way how they're asking us to do the accuracy, but they usually can accept whatever in the USP and EP. And if we go, okay, ICH is again, it's up to us to perform the accuracy the way we want it, but USP and EP are mentioning microscopy. And here comes the challenge. Microscopy, how exactly to perform it with microscopy? Is it with standard? Is it with the same sample? Or, both of them. So usually both of them is coming together using a standard and using your sample with the microscopy. And then ISO, that is trueness in ISO, but it's again mentioning orthogonal method such as microscopy. And what we did is we went out for all the R&Ds. We have a global forum. So we brought the experts from all the sites, PSD experts, and we asked them, okay, how do your site do accuracy? And we found 3 different methods. So 3 different methods in lots of different sites. So basically, 3 of them are correct because all these methods were submitted in one authority or another, and it was accepted. So what we decided to do is to take all these 3 methods and to write a white paper summarizing the 3 approaches and send this to all other sites. This is the way that you can basically select, depends on the technology that you have, depends on the particle size that you have, or the standards that's available. And it is acceptable so far, so we are happy with it. But from this, we came to I think that was also raised yesterday, we came to the idea that there should be a global working group on particle size that will bring together the authorities, the FDA in this case, the industry, and the system manufacturers. And if we work together with the main, painful topics, and it was so clear yesterday in the discussion that I noticed that, if you take question number 1 from yesterday, and you go for the tables that deal with question number 1, most of them have the same, each question have 4 topics. So most of them selected the same 2 topics in all the tables. It was so obvious that the pain point is here for everybody. So these are the things that we can work with globally. And I hope that one of the consequence of this meeting that we create such a working group. Setting specification is very painful, specifically for the generic industry, not only because we acquire the drug substance. So usually we do not manufacture the drug substance. We acquire it from a different company, and they have their method, and they need to set the specification, and both of us need to meet the same specification. It's not acceptable that they have spec and we have another spec, even though there's some time change between when we test the sample and when they test the sample. And as I said, so it is very important that the specification should include their spec, our spec that we, in the R&D, we test the sample a few months after that, and then the manufacturing site spec, which is another method. So we have 3 sites who have basically the same method. It's not usually the same method, but similar method, testing the same material, and we usually see different results, but we need to sit within the same specification. And as I said, 3 samples, this is what we have, at least in the beginning. So what we think there should be a process that we can, post-approval, submit specification change according to more and more batches that we have, but that process is still a little bit long and painful. And the last topic that I think it was raised yesterday, but this is just to finalize, is a software validation. Whenever there is a new software version, we face lots of problems because we need to revalidate the 21 CFR Part 11 validation of the system, and that will take a long time. And this is the expectation. I think it's time to change the expectation that whenever the software update is not including any change to the 21 CFR Part 11, a justification will be good enough. And with this, I will thank you and thank you.
Thank you, Dr. Ayub, for that great talk and introducing us to the tools, AI tools that your company is implementing. With that, I'd like to invite Dr. Dominic from Teva for the next presentation.
Good morning. I'll be presenting on the hurdles, challenges with regulatory submissions. And this comes from the generic industry perspective. My name is Bernard, and I'm a Director of Regulatory Affairs at Teva Pharmaceuticals, Inc. A short disclaimer here. The information provided here is mainly my research, and it's not necessarily the views of Teva. And the information provided is for educational purposes. Okay, here's a short agenda. I'll be talking about particle size and why it matters. And we'll take a look at the questions to understand what the issues are and the challenges. And from these FDA questions, I'll summarize at a high level what the pain points are. And we'll also take a look at the resources that are available to address these challenges or even mitigate it. And once we look at the resources, then we'll again go back and take a relook at the hurdles and challenges. Okay, so as we talked yesterday and we'll continue to talk today, particle size is important for drug development as it impacts both solubility, dissolution, bioavailability, and stability and product performance. The particle size also impacts dissolution, which again is critical for PCS class 2 and class 4 drugs. And in formulation performance, powder flows, settling, redispersion, etc., and stability. And in inhalation and nasal delivery, it is very critical for drug delivery at the target site within the respiratory system. And also for us regulatory folks, it is very important because in the post-approval world, we don't have a lot of equivalent studies carried out for major changes. And at this time, we require the particle size for comparison to what was previously approved and what we are changing with, especially to support an IV in vitro, in vivo correlation, as well as the PBPK studies, the physiology-based pharmacokinetics. So let's look at the questions. So I just compiled, I took a sample pool of 280 letters from both FDA and Teva. So 203 from FDA's website and 77 from Teva. And this is the breakdown. So we had the largest number of questions from specifications, and then you can go down. And the next was related to data, clarification, justification. And thankfully, method validation comes very low in this order. And we had some miscellaneous questions, which are all over the place, having partly from specifications, method validation, and so on, and the questions related to documentation. And so this is how the questions look like. So diving deep, going into the specifications, the most frequently asked questions was to establish a 3-tier specification for drug substance excipients, especially intermediates, where, for example, the pellet technology, and for the drug product. Okay, and also the questions on PSD control limits. There is always a recommendation to revise based on the data presented. Again, revise the drug substance PSD based on DMF. And again, here we have a dilemma because sometimes you also get questions where they say tighten it based on your development batches. So again, do we go with the DMF or do we go with what we have in development? Add 3-tier specifications for dry powder as well as for the reconstituted suspension. Establish specification for DS, even though PSD is not critical due to high solubility. Since the drug substance load is low, this impacts the formulation, and therefore there's a request to introduce the PSD. Tighten the specification for drug substance since it is a class 2 or class 4 drug. Explain why there are specifications on micronized and micronized material, and provide rationale for new API PSD, which is close to the PSD of API batches used in failed BE studies, and explain wider limits. So the questions, after the specifications, the second most common questions were related to data. And in this, the most frequently asked question was data presented in different places in our application, which may be inconsistent, especially the development report, the in-process sections, also the drug substance sections, and for certain technologies, the finished product as well. So provide PSD data for the reconstituted suspension in addition to powder, comparative data for the test and references to drug for semi-solids, provide data to support the selection of mentioned horizontal distance and class slide angle. So although this was carried out, but the supporting data was not available. Data requested to support the effect of storage on particle sizing. And there's also other additional data requested, which is the trend data analysis, number of particles counted, and percentage of particles which are agglomerates. Show no growth of particles with time. And you also have this APSD data of RLD, a lot close to expiry. Okay, pre and post actuation data demonstrate no agglomeration and particle breakage. PSD data for whole time study. And provide comparative dose accuracy testing data between the proposed product and the RLD, supporting complete dose delivered without submicron particle formation events prior to administration, as provided in the labeling. So the DLS analysis may be used to determine submicron particles. So the next set of questions are related to the clarification justification, and here you have the most frequently asked question is the justification of specification, and also a reason for one-time study, and also clarify impact of PSD on variability, on dissolution and drug layering, justify parameters used, and clarify number of calibration curves for the APSD studies. Sensitivity of the drug to UV light. Please specify any precaution taken during APSD study. Clarify automated actuation system will be used for APSD study. If yes, submit validation data to support. Explain the impact of PSD on PK data results since some PK studies failed. I think that is kind of common. We have seen it repeated. Explanation requested for using 10 inhalation batches in APSD studies and why LOQ value is considered sufficiently low enough. Justify APSD mass balance, OS, APSD mass balance not within the label claim. Clarify discrepancy in the submitted data and / anomaly in results. More than 95% of particles were in the 1 to 5 micron size range. Provide rationale. The particles not deposit in the lung GIT, and this is typically seen for inhalation products. And results using updated methods differ significantly from the older method. Please explain. And we have seen this more happening when we change the instrumentation or move to an upgraded instrument. So next we'll talk about the method validation questions. And here the most frequently asked questions relate to method validation, USP equivalence, specificity, robustness, and in some instances also accuracy. Develop more precise, accurate methods for PSD control. Method validation data in terms of zeta average and polydispersity index. Provide acceptance criteria. Data in SAS format. And also demonstrate accuracy of morphologically directed Raman spectroscopy. There's also been questions on justifying the selected particle morphology filters for distinguishing API from excipient particles and agglomerates from single particle. Provide validation data to justify the use of circle equivalent diameter that is not blind to significant percentage of API particles. Weight residual from histogram of in-process data is over 1%, which may indicate incorrect use of refractive index and absorption values. Address the impact of different LOQ values in 2 different method versions on the study results. Provide additional complementary methods to DLS. Use reconstituted instead of diluted samples. Develop validated analytical methods suitable to characterize the aerosol drug particle size distribution, and provide evidence that the proposed conditions do not result in particle size reduction. So the next set of questions is related to document requests and miscellaneous. And here you'll see the most frequently asked questions is supporting validation documents and previous method, especially when there is a change in the method. And we see a lot of these questions in the post-approval world. There's also requests for SOP protocol and provide method used to avoid test and RLD bias and dose release of manual actuation. Method development report for particle size distribution of the constitutive suspension. Provided comparative physicochemical data do not support equivalency of the proposed drug product, the pivotal in vivo study and pivotal PSD study are not acceptable, and FDA is recommending new batch. The potential cause identified for OS of delivery of delivered dose uniformity and aerodynamic particle size distribution is not acceptable. Request to repeat the study. And the other common question is request for samples and material. Please provide drug product characterization studies as applicable to your drug product per guidance for industry. PSD study not acceptable. Repeat study with more than 1 measurement and adequate number of samples. Since proposed product does not comply with USP PSD test 2 standard, please demonstrate compliance to test 1. Your justification is not acceptable since monograph is available. So next we'll talk about the issues and challenges against the backdrop of the above FDA questions. So where are the challenges? Is it with the basic fundamentals, robust development? Is it with the validation? Is it with the methods and the specification setting? How about physical characterization? We have a guidance on Q3 with particle size distribution as one of the parameters. And what about, are we looking at data inconsistencies between what we actually develop and present in the PDR versus other sections of the ANDA? And with regards to development and validation, accuracy and selection of standards. Methods suitable to characterize. So are we actually looking out at our methods? So when you see validation challenges and corresponding skewed data, then does the finger point to the method suitability? How is the instrument contributing to the skewed data? And then are we also looking at complementary methods for complex products, especially in situations where not only the size is significant, but also the shape of the particles? So interpretation. Also, we have seen issues and challenges here between how FDA versus industry interprets the data that is presented. Some examples are impact of PSD on PK data results, use of LD versus DLS, or request for additional techniques data, drug product characterization outcomes, and this is seen mainly in semi-solids. So I'll talk about next the resources that are available to the industry and various stakeholders. We see that we have the guidance for industry, the ICH monographs, and product-specific guidances, etc. Okay, we have the physical, chemical, and structural characterization, which is the ICH Q3. We have the nasal spray and inhalation solution suspension and spray dry products, chemistry, manufacturing, and controls documentation. We have the guidance on oral drug products administered via enteral fluid tube, in vitro testing, and labeling recommendations. And this also talks about particle sizing required, especially for pellet products. Size of beads in drug products labeled for sprinkle. So this is the food effect study. Then the general principles for evaluating the abuse deterrence of generic solid opioid drug products. There's also the guidance on drug products, including biological products that contain nanomaterials. And some sections of the post-operal changes to drug substance also talk about particle sizing. Okay, we have the ICH Q13, which talks about continuous manufacturing of drug substances and drug products, with information also on particle sizing, and ICH Q6A. So the USP monograph is another treasure trove of general guidances. We have the USP 430 on particle size analysis by DLS, the 429 on LD measurement of particle size, the USP 1003 for resuspensibility and redispersibility, the 1430.2 for the analytical methodologies based on scattering phenomena, and you have the 1430.3 for the DLS and the 1430.2 for the LD. You also have the particle counting via light scattering, which is the 1430.6. The 1604 talks about presentation of aerodynamic particle size distribution measurement data for orally inhaled products. And 776 talks about optical microscopy. We also have certain, a lot of product-specific guidances from a couple of years back when we did not have a lot of guidances, especially for complex products. I think there's a lot of improvement in the guidances that are available that addresses particle sizing. And also, if you look at the upcoming product-specific guidances for generic products, that also holds a lot of promises, because a lot more guidances are also in publication, and hopefully it'll be out soon. So let's look at the aripiprazole Loroxil injectable suspension. It has a requirement for in vitro bioequivalence study with particle size distribution. The azacitidine also has a particle size distribution with a comparison using the population bioequivalence. And the budesonide formoterol fumarate inhalation aerosol talks about the population bioequivalence again with aerodynamic particle size distribution. And the rivaroxaban for suspension, it talks about in vitro feeding tube studies with particle size distribution. The oxycodone extended release has both the food effect study as well as the abuse deterrence studies, and each of this has its own particle size recommendations. And you would have seen previously, all of these PSG guidances also have the other FDA guidances that kind of complement each other. So going back to the hurdles, issues, and challenges, despite all these guidances and USP and ICH and product-specific guidances, what are the challenges faced by industry, and what is the way out of the challenges? So basically, it all boils down to our development story. Are we telling our development story? Are we talking about one technique versus the other? What are the difficulties in the method, and what are the statistical challenges? So in most times, while telling a development story, we do not emphasize where particle size fits, right? Most times because we have dissolution, we have bioequivalence, we have other parameters like viscosity, dispersion, and so on. do we push PST into the back burner, only to realize, after, approval, when we have challenges, then we realize how important PST becomes, right? So were any IVC correlations, PT, IVRT, etc., considered? And the Q3 guidance on characterization includes PST as a very important parameter. And therefore, is it adequate to support your development story and satisfy the reviewer? And also, do we have a good understanding of PSG recommendations with regards to the first principle-based technique? So despite the above resources, the industry also deals with impractical PSD limits based on limited number of batches, conditioning review of development validation with the experiences from multiple other products, development validation that reviewers are exposed to. So we have seen this issue where we present a story in the development report, but then we start getting questions. And we see that there is, reviewers are exposed to a lot of information, to a lot of applications. And therefore, we begin to wonder, are they clouded by all this information? Because it's a wealth of information, but then if you're not focusing on the specific application, the technology used, the development story, then are you all biasing your review towards, a generalized view instead of the specific application? And also the one-size-fits-all phenomenon. So is this is a particular review comment imported from another product, right? And also there's also the other phenomenon we have seen where although we have a good development story and the data aligns and from end to end there is certain rationale, we still see that there are questions. And then we begin to wonder, again, since FDA reviewers themselves are SMEs and they have a lot of information and a lot of resources available to carry out their review, right? So in this case, are they critiquing the PSD development? And so you have this situation where the author knows better, so the critic knows better than the author. And also, so we really need FDA to start looking at the method, the development, and so on from the developer validator's lens. Okay, and also there's scope for improvement on the PSGs. Of course, we do understand that not every product has a PSG. And also PSGs don't address PSD for complex products. Okay, and also we see regular PSD challenges in relation to FDA guidances of PSD recommendations, and that is like a practical issue. So what more can industry do in addition to robust development validation? Okay, so FDA gives us a lot of avenues here. So you can leverage your pre-development NDA meetings to tell your story and ask questions. Participate in requests for public comments to PSGs. So when in doubt, submit control correspondences. Okay, and also utilize mid-cycle review meetings or post-CRL meetings. Okay, and as you present your development story, talk more about the challenges and get a buy-in. So you can use all of these forums to, to present your story and to get your buy-in. Okay, with this, I conclude my presentation. Thank you, everyone.
Thank you, Mr. Dominic, for that great presentation and for some suggestions on ways out. With that, we conclude session 6. We're going to take a short bio break for the people who are in person. I know we're running behind schedule, so we'll take a 10-minute break. We'll be back at 10:55 to get started with session 7, where we'll have the vendor's perspective on laser diffraction. Thank you, everyone, and thank you to everyone who's online.